Welcome to our deep dive into the tumor microenvironment, complex and dynamic space where cancer cells interact with their surroundings. This environment is not just a backdrop, it plays an active role in tumor growth, progression, and response to treatment. At the heart of the microenvironment are two key players: the cellular components, which include various types of cells, and the extracellular matrix, or ECM, which provides structural and biochemical support. Understanding how these elements work together is crucial for developing new cancer therapies. In this video, we'll explore the fascinating interplay between cells and the ECM within tumors and why this relationship matters. Extracellular matrix, or ECM, is a network of proteins and carbohydrates that surrounds and supports the cells within tissues. Think of it as a scaffold that not only holds cells in place but also influences their behavior. In the context of tumors, the ECM is often altered, becoming denser or stiffer, which can affect how cancer cells grow and spread. The ECM is made up of components like collagen, elastin, and glycoproteins, each playing a unique role in maintaining tissue structure and signaling to cells. Understanding the ECM is key to unraveling how tumors develop and resist treatment. Within the tumor microenvironment, a variety of cells interact with each other and with the ECM. These include not only cancer cells but also fibroblasts, immune cells, endothelial cells, and more. Each cell type has a specific function, from supporting tumor growth to attempting to fight it. The balance and communication between these cells can determine how aggressive a tumor becomes. For example, cancer-associated fibroblasts can remodel the ECM, while immune cells might be suppressed by signals from the tumor. This cellular diversity makes the tumor microenvironment a highly dynamic and challenging space. Cancer cells do not exist in isolation. They constantly communicate with neighboring cells, exchanging signals that can promote or inhibit tumor growth. These interactions can lead to changes in the ECM, making it easier for cancer cells to invade surrounding tissues. Additionally, cancer cells can recruit other cell types like fibroblasts and immune cells to support their survival and spread. This network of communication is a key reason why tumors are so difficult to treat, as the microenvironment can shield cancer cells from therapies. Fibroblasts are a type of cell found in connective tissue, and in tumors, they often become activated as cancer-associated fibroblasts, or CAFs. These cells play a major role in remodeling the ECM, producing proteins like collagen that can stiffen the matrix. This stiffening can create a physical barrier to immune cells and therapies, making it harder to treat the tumor. CAFs also secrete growth factors and cytokines that support cancer cell survival and invasion. Their activity is a double-edged sword, sometimes helping to contain the tumor but often aiding its progression. Immune cells are another crucial component of the tumor microenvironment. They can recognize and attack cancer cells, but tumors often develop ways to evade or suppress the immune response. Some immune cells, like T cells and natural killer cells, are directly involved in targeting cancer. Others, such as macrophages, can be co-opted by the tumor to promote growth and suppress other immune responses. The balance between these immune cell types can influence whether a tumor is controlled or allowed to progress. Epithelial cells line the inside of blood vessels and are essential for forming new blood vessels, a process called angiogenesis. In tumors, angiogenesis is often hijacked to supply the growing mass with nutrients and oxygen. However, these new vessels are usually abnormal, leading to areas of low oxygen or hypoxia within the tumor. This can make the tumor more aggressive and resistant to treatment. Targeting angiogenesis is a strategy used in some cancer therapies, aiming to cut off the tumor's lifeline. ECM provides more than just a physical scaffold for cells. Its structure can influence how cells move, grow, and interact. In tumors, changes in the ECM's composition and organization can create pathways for cancer cells to migrate or barriers that block immune cells. The stiffness and density of the ECM are often increased in cancer, which can promote tumor progression and make it harder for drugs to penetrate the tissue. Understanding these structural changes is key to developing better treatments. Beyond its structural role, the ECM acts as a signaling platform, influencing cell behavior through biochemical cues. Proteins in the ECM can bind to receptors on cell surfaces, triggering pathways that control cell growth, survival, and movement. In cancer, these signals are often hijacked to promote tumor progression. For example, certain ECM components can activate pathways that help cancer cells resist cell death or become more invasive. This signaling function makes the ECM a target for innovative cancer therapies. Collagen is the most abundant protein in the ECM and plays a central role in maintaining tissue structure. In tumors, collagen fibers can become densely packed and aligned, creating tracks that cancer cells use to invade surrounding tissues. This reorganization of collagen not only supports tumor growth, but also makes it more difficult for immune cells and therapies to reach the cancer. Targeting collagen remodeling is an area of active research in cancer treatment. Elastin is another important ECM protein, providing tissues with elasticity and flexibility. In the tumor microenvironment, changes in elastin levels can alter the mechanical properties of the tissue. This can influence how cancer cells move and how the tumor responds to physical forces. Reduced elastin can make tissue stiffer, while increased elastin may help tumors adapt to their environment. Understanding elastin's role could lead to new ways to target the tumor microenvironment. Glycoproteins are ECM components that play a key role in cell adhesion and communication. They help cells stick to the ECM and to each other, influencing how they move and interact. In cancer, changes in glycoprotein expression can promote tumor cell detachment, invasion, and metastasis. Some glycoproteins also act as signaling molecules, affecting cell growth and survival. Targeting glycoproteins is a promising strategy for disrupting tumor progression. Proteoglycans are large molecules in the ECM that help retain water and maintain tissue hydration. In tumors, altered proteoglycan levels can affect the movement of cells and molecules through the ECM. This can influence how cancer cells invade and how drugs are delivered. Proteoglycans also interact with growth factors, modulating their availability to cells. Their role in ECM hydration and signaling makes them important players in the tumor microenvironment. Matrix metalloproteinases, or MMPs, are enzymes that break down ECM components. In tumors, MMPs are often overactive, leading to excessive ECM remodeling. This can create pathways for cancer cells to invade and metastasize. MMPs also release growth factors stored in the ECM, further promoting tumor progression. Inhibiting MMP activity is a potential strategy for limiting cancer spread, though it comes with challenges due to their complex role. One of the most significant changes in the tumor microenvironment is increased ECM stiffness. This physical property can promote cancer cell invasion and resistance to therapy. Stiffer ECM can activate signaling pathways that make cancer cells more aggressive and less responsive to treatment. Researchers are exploring ways to target ECM stiffness as a means to improve cancer therapy outcomes. Hypoxia, or low oxygen levels, is common in tumors due to abnormal blood vessels and dense ECM. Hypoxic conditions can make cancer cells more aggressive and resistant to therapy. The ECM contributes to hypoxia by limiting blood flow and oxygen diffusion. Understanding how hypoxia and ECM changes interact is important for developing new treatment strategies. Dense and stiff ECM in tumors can act as a barrier to drug delivery. Therapeutic agents may have difficulty penetrating the ECM to reach cancer cells, reducing their effectiveness. Researchers are developing strategies to modify the ECM or use specialized drug carriers to overcome this challenge. Improving drug delivery through the ECM is a key focus in cancer therapy research. Cancer cell invasion and metastasis are major challenges in cancer treatment. The ECM plays a critical role in these processes by providing tracks for cancer cells to move and by being remodeled to allow passage. Enzymes like MMPs break down ECM barriers, while changes in ECM composition can promote cell detachment and migration. Targeting these mechanisms is essential for preventing cancer spread. Tumor-associated macrophages, or TAMs, are immune cells that often support tumor growth instead of fighting it. They can promote ECM remodeling, suppress other immune responses, and help cancer cells evade detection. TAMs are a major focus of research, as targeting their activity could shift the balance in the tumor microenvironment toward anti-tumor immunity. Cancer-associated fibroblasts, or CAFs, are specialized fibroblasts found in the tumor microenvironment. They produce ECM components, secrete growth factors, and influence immune cell behavior. CAFs can create a supportive niche for cancer cells, making them more resistant to therapy. Targeting CAFs and their interactions with the ECM is a promising area for new cancer treatment. Tumors use various strategies to evade the immune system, including altering the ECM to create physical barriers and secreting molecules that suppress immune cell activity. These changes can prevent immune cells from reaching and attacking cancer cells. Understanding immune evasion mechanisms is critical for developing effective immunotherapies. Angiogenesis, the formation of new blood vessels, is essential for tumor growth. The ECM provides signals and structural support for angiogenesis, while endothelial cells build the new vessels. However, these vessels are often abnormal, leading to inefficient blood flow and hypoxia. Targeting angiogenesis is a strategy to starve tumors and limit their growth. Cancer stem cells are a subpopulation of cells within tumors that can self-renew and drive tumor growth. The ECM provides a niche that supports these cells, protecting them from therapy and promoting their survival. Disrupting the ECM cancer stem cell interaction is a potential strategy for preventing tumor recurrence. Mechanical forces within the ECM, such as tension and compression, can influence cancer cell behavior. Stiffer ECM can activate signaling pathways that promote invasion and resistance to therapy. Understanding how mechanical forces shape the tumor microenvironment is important for developing new treatment approaches. Chronic inflammation is common in the tumor microenvironment and can promote cancer progression. The ECM can influence inflammatory responses by interacting with immune cells and releasing signaling molecules. In turn, inflammation can lead to further ECM remodeling, creating a feedback loop that supports tumor growth. Tumors are highly heterogeneous, meaning they contain a diverse mix of cells and ECM components. This diversity can make tumors more adaptable and resistant to therapy. The ECM contributes to this heterogeneity by providing different microenvironments within the tumor, each supporting distinct cell populations. Some cancer cells can enter a dormant state, remaining inactive for long periods before reactivating. The ECM can influence dormancy by providing signals that keep cells in a quiescent state. Understanding how the ECM regulates dormancy could help prevent cancer recurrence after treatment. The ECM can contribute to therapy resistance by creating physical barriers, activating survival pathways, and supporting resistant cell populations. Overcoming ECM-mediated resistance is a major goal in cancer research, with strategies including ECM modification and combination therapies. Targeting the ECM is an emerging strategy in cancer therapy. Approaches include inhibiting ECM remodeling enzymes, modifying ECM stiffness, and disrupting cell-ECM interactions. These strategies aim to make tumors more vulnerable to existing treatments and prevent progression. Immunotherapy is a promising cancer treatment, but the ECM can limit its effectiveness by blocking immune cell access and promoting suppressive signals. Combining immunotherapy with ECM targeting strategies may enhance treatment outcomes and help overcome resistance. Nanotechnology offers new ways to deliver drugs through the ECM. Nanoparticles can be designed to penetrate the dense matrix and release therapeutic agents directly to cancer cells. This approach holds promise for improving drug delivery and reducing side effects. The ECM contains biomarkers that can be used to diagnose cancer, predict prognosis, and monitor treatment response. Identifying and targeting these biomarkers is an active area of research with the goal of personalizing cancer therapy. Researchers use 3D models to study the tumor ECM and its interactions with cells. These models better mimic the complexity of real tumors compared to traditional 2D cultures, providing valuable insights into tumor biology and therapy response. Animal models are essential for studying the tumor microenvironment and testing new therapies. They allow researchers to observe how the ECM and cells interact in a living system, providing insights that guide the development of new treatments. Personalized medicine aims to tailor cancer treatment to each patient's unique tumor microenvironment, including the ECM. By analyzing ECM composition and cell interactions, doctors can select therapies that are more likely to be effective for individual patients. The field of ECM research is rapidly evolving, with new technologies and discoveries shaping our understanding of the tumor microenvironment. Future directions include developing better models, identifying new therapeutic targets, and integrating ECM analysis into clinical practice. Despite progress, targeting the ECM in cancer therapy presents challenges. The complexity and diversity of the ECM, as well as its essential roles in normal tissue, make it difficult to develop safe and effective treatments. Ongoing research aims to overcome these obstacles. The ECM can contribute to tumor recurrence by supporting dormant cancer cells and creating a protective niche. Targeting ECM components involved in recurrence is an important strategy for improving long-term cancer outcomes. The composition and role of the ECM can vary between different types of cancer. Understanding these differences is important for developing targeted therapies and predicting how tumors will respond to treatment. Patient-derived ECM models use tissue samples from cancer patients to study the unique features of their tumor microenvironment. These models help researchers understand individual differences and test personalized treatment strategies. The ECM can influence tumor immunity by affecting immune cell infiltration and function. Modifying the ECM to enhance immune responses is a promising approach for improving immunotherapy effectiveness. The ECM can influence tumor metabolism by regulating nutrient availability and waste removal. Changes in ECM composition can affect how cancer cells obtain energy and adapt to their environment, impacting tumor growth and therapy response. The ECM plays a role in tumor evolution by shaping the selection pressures that drive genetic and phenotypic changes in cancer cells. Understanding how the ECM influences tumor evolution can inform strategies to prevent resistance and recurrence. Summary: the tumor microenvironment is a complex network of cells and extracellular matrix components that interact to influence cancer progression, therapy response, and patient outcomes. By understanding the roles of both cellular and ECM elements, researchers and clinicians can develop more effective, targeted cancer treatments. The future of cancer therapy lies in unraveling these intricate relationships and translating discoveries into clinical practice. As research into the tumor microenvironment continues, the interplay between cellular and extracellular matrix components remains a key focus. Advances in technology and a deeper understanding of these interactions hold the promise of new, more effective cancer therapies. By targeting both the cells and the ECM, we can hope to overcome current challenges and improve outcomes for patients worldwide. Thank you for joining us on this exploration of the tumor microenvironment.